So now the question is, what is Nemec phenomenon? So how this Nemec phenomenon is related? So we see this Nemec phenomenon in embryology. In embryology, what we see, the structure of the male gametophyte, it just appears like female gametophyte or the embryo sac. See, if you see the difference between the male gametophyte, that means the structure of the microspore. If you see the structure of the microspore, we see the exine, intine, and a single nucleus in the microspore. That is also called as male gametophyte or pollen grain. But Nemec, he observed that there is a embryo sac-like structure in the pollen grain. Like instead of a single nucleus, maybe that single nucleus may be divided into two nuclei, tube nucleus or vegetative nucleus. Another one is called generative nucleus. Maybe we can find two nuclei, but in this pollen grain, we find 8 nuclei, just like in case of the female gametophyte or embryo sac. So this phenomenon is called as Nemec phenomenon. So now the options are, what is Nemec phenomenon? Options are development of pollen grain embryo sac from microspore mother cells, development of pollen grain like embryo sac from microspore mother cell. So that is the right answer. Okay, pollen grain, it appears like embryo sac. That is the right answer. And other options are given: entry of pollen tube into ovule through micropyle, that is called as porogamy. That is not the right answer. Entry of pollen tube into ovule through chalaza, that is called as chalazogamy. That is also not the right answer. And entry of pollen tube into ovule through funiculus. So that is not also the right answer. So the right answer is Nemec phenomenon is related to the appearance of the pollen grain, like just like an embryo sac. Okay, that is the right answer. Next one is, in which type of ovule the micropyle comes close to the base of funiculus? In which type of ovule the micropyle, it comes close to base of. Now we see the different types of the ovules. You see, this is the atropous or anatropous ovule. So this is the funiculus and this is the micropyle. Both are opposite to each other. Funiculus, chalaza, and micropyle, they are opposite to each other. So it is not the right answer. If you see the anatropous ovule, see, this is the funiculus and this is the micropyle. And in case of the amphitropous ovule, you see, this is the funiculus, this is the micropyle. And in case of the hemianatropous ovule, this is the funiculus, this is the micropyle. And campylotropous ovule, funiculus and micropyle. And succinotropous ovule, here it is just like spring, it formed a spring-like structure. So where we find both the funiculus and micropyle in a very close structure. Both are arranged very closely. So you see, in case of the amphitropous, it might be little closer, and in case of the campylotropous also it is closer. But in case of the anatropous ovule, we see, because it is the ovule body in the inverted position. Okay, in the anatropous ovule or atropous ovule, in the arthropous ovule or the atropous ovule, it is the straight. The ovule body is arranged straight. That is 0 degrees. But in anatropous ovule, the ovule body with reference to funiculus, it is just inverted. It is arranged in 180 degrees angle. In the anatropous ovule, the ovule body is 180 degrees angle with reference to funiculus. So this is the right answer. Anatropous ovule is the right answer. So దీన్ని తెలుగులో వక్రాండం అంటారు. In which type of ovule the micropyle comes close to the base of funiculus means anatropous ovule. Both are very close, both micropyle and funiculus. And next question is monosporic 8 nucleate embryo sac that develops from chelagel megaspore is seen in. That is the question. Monosporic 8 nucleate embryo sac. So we see in the types of embryo sac development or the female gametophyte development, there are 3 types of the female gametophyte developments. One is monosporic, another is bisporic, and third one is tetrasporic. Okay, so here monosporic 8 nucleate embryo sac that develops from chelagel megaspore is seen in. So now they are asking, in the monosporic type of the embryo sac, there are 2 types. One is polygonum variety, another is Enothera variety. Okay, Elium, it is related to bisporic, and Peperomia related to tetrasporic type of the female gametophyte developments. So now we see the different types of the embryo sac here. One is 8. Yeah, here you see the monosporic 8 nucleate polygonum type. In the monosporic, there are 2 varieties, 2 types. One is polygonum type, another is Enothera type. Just they are asking only about the monosporic type. So now it might be polygonum type or Enothera type. Now what they are asking here, in case of the polygonum type, first megaspore mother cell, it undergoes meiotic division, meiosis 1 and meiosis 2. So here they form, in the meiosis 1 it formed 2 megaspores, and after meiosis 2 it formed 4 megaspores. The upper one is called as micropylar end, the lower one is called as chelagel end. So here in the polygonum type, the megaspores which are formed, there are 4 megaspores. Out of 4 megaspores, the 3 megaspores which are at the micropylar end, I told you, no, the upper one is called as micropylar, lower one is called as chelagel end. So those 3 megaspores, sorry, which are arranged at the micropylar end, they degenerate, and the megaspore, the 4th megaspore, which is located at the chelagel end, it remains active, that is functional megaspore. And in case of the Enothera type variety, it is just opposite to. In the Enothera variety, the megaspore which is at the micropylar end is functional. So the question is, in which of the monosporic type of the embryo sac we find the chelagel megaspore functional? So the answer is polygonum. But because in the polygonum type, the megaspore which is located at the chelagel end is functional. So that is the right answer. Okay. In the monosporic 8 nucleate embryo sac that develops from chelagel megaspore is seen in. So that is the polygonum is the right answer. Okay. And Enothera, it is quite opposite to this polygonum. And Allium, it is not at all related to monosporic, it is bisporic. And Peperomia also, it is not at all related to monosporic, it is a tetrasporic embryo sac. Next, the question is, Which of the following is the DNA model of Watson and Crick? So actually, Watson and Crick, they initially proposed the DNA model. Okay, that is a double stranded helical structure. Okay, so here there are later 3 different types of the DNAs are observed. One is aDNA, another is bDNA, and next one is zDNA or gDNA. So now the DNA we know is bDNA. Okay, and aDNA is right-handed. So you should know the differences between the aDNA, bDNA, and zDNA. aDNA and bDNA both are right-handed. They form the right-handed helical structure. They turn in the right direction. Those helical structures turn in. It is a double helical structure. Those helical structures turn in right direction. And zDNA or zDNA, it is a left-handed structure that you should know. Okay, and you see major group. In case of the bDNA, it is narrow, and in case of the aDNA, it is wide, deep. That is the main difference between the aDNA and bDNA. And so the right answer, what is the right answer? Which of the following is the DNA model of Watson-Crick? So what is the DNA model proposed by the Watson-Crick? That is the bDNA. This is the normal DNA we find in all the most of the organisms. And later they found aDNA and zDNA. zDNA is left-handed structure, so it is not the one. So and aDNA is a right-handed structure. Next one is, which non-genetic RNA is the major RNA present in the cell? Okay, in the cell there are 3 types of RNAs are present. One is mRNA, tRNA, and rRNA. Messenger RNA, transfer RNA, and rRNA, that is the ribosomal RNA. snRNA, it is not related to this. This is a small nuclear RNA. It is a different one. And so mRNA, it is a genetic RNA because it is transcribed from the directly from the DNA, and it is useful for the translation of the proteins. And which one is the major RNA present? That is the ribosomal RNA, because the RNA which is present in the ribosomes is called as ribosomal RNA. For example, in the 7-case ribosomes, there is 16S rRNA. Okay, 16S rRNA and 5S rRNA, they are present. So they are, because each cell has many ribosomes. Ribosomes are very important for the translation. So in the ribosomes there is much we find rRNA. It is present abundantly in the ribosomes because ribosomes are constructed, made of proteins, ribosomal proteins, and ribosomal RNA. So that is the answer. Ribosomal RNA is the right answer. It is a major RNA present in the cell. Next question is RAN-guanine exchange factor. It is also called as RAN-GEF or present in. Where we find RAN-GEF? So that is the question. Usually it is not, it is very deep question. Okay, it is this guanine exchange factor. It exchanges between the GTP and GDP. If you see here, RAN-GTP regulates nucleocytoplasmic transfer. That means this RAN-GF, RAN guanine exchange factor, it is present in the nucleus and it exchanges between the RAN GDP and RAN GTP. And also this RAN GEF plays an important role in the cell division. It determines the cell division. It regulates the cell division. So the answer, RAN GEF are present in, where they are present? They are present in the nucleus. They exchanges the GTP and GDP with the GTP in the nucleus. That is required for the translation. Okay, next. What is Chargaff rule? Chargaff rule, it is related to DNA structure. What Chargaff proposed? See here, nucleotides are formed by either DNA or RNA. So it is not the Chargaff rule. And nucleotides are formed by both nucleic acid. That is also not the Chargaff rule. Purines and pyrimidines are present equally in DNA. Exactly. This is the right answer. As per the Chargaff rule, both the purines and pyrimids are in equal proportion. Because one purine always forms hydrogen bonds with the one of the pyrimidines. That means AT. Adenine is the purine. It forms two hydrogen bonds with the T. That is the thymine. Thymine is the pyrimidine. And guanine, it is the purine. It forms three hydrogen bonds with the C. That is cytosine. That is pyrimidine. So both the purines and pyrimidines are in 1:1 ratio. Both are equally present in the DNA. That is the rule proposed by the Chargaff. So it is called a Chargaff rule. Next one is, which statement is not correct regarding DNA and RNA polymerases? See, there are DNA polymerase and RNA polymerase also in the cell. So DNA polymerase are required for the DNA replication. It is also called as autocatalysis. Okay, DNA acts as a template itself for the DNA synthesis. That is called as DNA replication or semi-conservative replication. You see here, in the DNA, DNA is double stranded, and both the DNA strands, they separate from each other during the replication. DNA polymerase is required for the replication, and for the replication, a primer is also required. See, this is the template of the DNA, 3' to 5'. So this primer finds at the 3' strand of the template, and this primer is required for the replication of the DNA with help of the DNA polymerase. Okay, both the strands are separated and they act as the templates. And the DNA synthesis, it occurs from 5' to 3' direction. So it requires primer. And if you see the RNA polymerase, it does not require a primer. Okay, so here there are some differences between the DNA dependent DNA polymerase and RNA dependent RNA. That is called as RNA polymerase. Okay, DNA polymerase and RNA differences. Catalyzes DNA template directed extension of the 3' end of the DNA strand. From the template of the 3' end, the DNA synthesis starts from 5' to 3' direction. Okay, so here, in case of the, you see the options. Which statement is not correct regarding DNA and RNA? Not correct. You should be very clear about this, the question. What they are asking is, out of the 4 options, which is not correct? Now we see the options. Both enzymes, that means DNA polymerase and RNA polymerase, add nucleotides to the 3' end of each. Yes, that is the right answer. But here they are asking about the not correct, which is not, that means they are asking about the not correct option. So the first one is right. They add nucleotide at the 3' end, both of them. And second option is both enzymes require a primer. Just now we discussed that DNA polymerase require a primer for the synthesis of the DNA, but RNA polymerase does not require a primer for the RNA synthesis. DNA polymerase synthesizes the DNA. RNA polymerase synthesizes the RNA with the help of DNA template. So only DNA polymerase requires the primer. RNA polymerase does not require the primer. So this is the option we have to keep, because they are asking about the not correct option. And remaining two are also right. DNA polymerase is used in replication, while RNA polymerase is used in transcription. Okay, that's we know. DNA polymerase is required for the replication of the DNA, and RNA polymerase is required for the transcription of mRNA. So that is also right. And the binding of RNA polymerase is preceded by the binding of the transcription factor. The binding of RNA polymerase is preceded by the binding of the transcription factor. That is also right answer. So since they are asking for the wrong answer, wrong one, we have to choose the second one. DNA polymerase requires the primer, but RNA polymerase does not require the primer. So that is the option we have to keep. Next, which of the following is not a correct statement regarding polytene chromosomes? Again, we are discussing about the chromosomes, polytene chromosomes. Earlier, we discussed lamp brush chromosomes and polytene chromosomes. Polytene chromosomes were discovered by Balbiani. Okay, and also they are found in the Drosophila. Okay, salivary glands. So here, what they are asking, again, this is the question related to not a correct state. That means they are asking to choose the wrong statement out of the four. Conspicuous banding is a special quality of polytene chromosomes. Conspicuous, that means very clear banding. In the polytene chromosomes, we see the bandings. They are also called as Balbiani rings. Okay, we see the banding pattern, very clear banding pattern in the polytene chromosomes. So that is the right option. That is the right statement. So we should not choose this one. And the bands on polytene chromosomes are DNA rich sections. That is also right statement. We see the bands wherever DNA is very rich. And third one is there is a correlation between band number and chromosome number. So this is the wrong statement. There is no relation between the band number and the chromosome. And chromosome number varies from organism to organism. And fourth one, no correlation between band number and the total DNA content in the genome. So this is also right statement. So third one is the wrong statement with reference to polytene chromosomes. So here we see the polytene chromosome, how they appear. We see the bands, the banding pattern. There is no correlation between the number of chromosomes and the number of bands. Okay, so that is the option we have to keep. There is a correlation between band number and chromosome. That is the wrong statement. So we have to choose the wrong statement. They are asking for the wrong statement or incorrect statement. Next, which one is a heterocystous nitrogen fixing aerobic cyanobacterium? So it is just related to nitrogen fixation. The cyanobacteria are blue-green algae. There are Trichodesmium, Oscillatoria, Anabina, and Pharmidium. Out of these four, only Anabina has the heterocysts. Heterocysts are the special structures which are present in the Anabina and Nostoc. but they are not present in the Oscillatoria, Trichodesmium, and Formidium. So the right answer is Anabina. It has heterosis, which are useful for the nitrogen fixation. And next one is, which cyanobacterial symbiont in Azolla makes it a good biofertilizer in rice cultivation? It is also related to cyanobacteria or blue-green algae. Azolla is an aquatic pteridophytic plant used as a biofertilizer, particularly in the paddy or rice cultivation. Azolla is an aquatic floating. It floats on the water. It is a pteridophyte. So it forms a symbiotic relation with the Anabina. The Anabina which is present in the Azolla is called as Anabina azollae. That is the species of Anabina. So Azolla has a symbiotic relationship with the Anabina. So there is a symbiotic relationship between the Anabina and Azolla. So that is the right answer. Not the Oscillatoria, not the Trichodesmium and Alocera. These are the wrong option. So you should remember, Anabina is the blue-green algae or cyanobacteria which resides, which forms a symbiotic relationship with the Azolla. And next, which type of plants initiate the zero-year succession? So you see in the succession, in the ecology, there are two main ones. One is hydroseer, another is xeroseer. Sometimes it is also referred as a lithoseer. The plants which grow on the rocks, surface of the rocks, are called as lithophytes. It was asked in the earlier question paper. Lithophytes are the plants which grow on the surface of the rocks. So here the xeroseer, it starts on the surface of the rocks. Okay, so here they are also called, the plants which start their life on the rocks are called as pioneers of the xeroseer, or pioneers of the succession, ecological succession. And here we see lichens. We see there are two types of lichens here given as the options. One is crustose lichens, another is a foliose lichens. The right answer is crustose lichens, because lichens start their life on the surface of the rocks, and crustose lichens are very thin structures, very thin. They can withstand, they can grow on the surface of the rocks, because lichen is a symbiotic relationship between the algae and fungi. The algal partner, it synthesizes the food with the help of by the photosynthesis, and fungal partner, which absorbs the water in the form of the moisture from the atmosphere. Since they are able to grow on the surface of the rocks, their roots cannot penetrate, since there is no soil. They cannot absorb the water with the help of the roots. Since they form the symbiotic association, the fungal partner, it absorbs water in the form of the moisture from the atmosphere, and it provides that water to the algal partner, and algal partner synthesizes the food by photosynthesis, and both of them share the food. So it is called a symbiotic relationship. So here the right answer is crustose lichen. They are called as pioneers of the ecological succession. They start this one growing on the rocks, and later they are replaced by foliose lichens. Okay, so next one is foliose lichen. The right answer is crustose lichen. And next one is, which of the following is a primary consumer? This is also from the ecology. So here in the food chain, we see the producer. Producers are always photosynthetic organisms. They provide the food to the consumers. Producers are the plants, algae. All these are the photosynthetic organisms. They are called as producers. And consumers, are mainly three types: primary consumers, secondary consumers, and tertiary consumers. And also there are also micro consumers and mega consumers. So now if you see the primary consumer, which one is primary consumer? Which directly depend on the consumers, which directly depend on the producers, are called as primary consumers. They are also called as herbivores because they depend on the plants. The primary consumers depend on the plants, just like deers, all these grasshoppers. They directly depend on the producers, that means plants. So they are called as herbivores. Next. Who introduced the progressive evolution in sporophyte in bryophytes? Which is very important. Okay. Evolution of sporophyte in bryophyta and stellar evolution in pteridophyta. These are most important topics. With reference to evolution of sporophyte in bryophyta, who proposed? There are two theories. One is progressive evolution theory, another is retrogressive evolution theory. Who proposed progressive evolution theory? Bauer. Bauer proposed the progressive evolution theory. That is the right answer. Kashyap, here it is a wrong answer. Sivaram Kashyap is an Indian botanist, but he proposed the retrogressive evolution of sporophytes. Okay, if you see the progressive evolution theory, because they may ask different questions on this. Rixia, it is very primitive bryophyte, and next Markamtia, next Pellia, next Anthoceras and Funaria. So if you see, Funaria is an advanced bryophytic. Here, if you see the sporophyte in Rixia, we see most of the sporophyte contains the sporogenous tissue. Okay, that is called fertile tissue. If you keep on looking from Markamtia to the Funaria, the content of the sterile tissues increase, keep on increasing. The fertile tissue content is keep decreasing with reference to sterile tissue. So, what is the purpose of sterile tissue? Is it progressive or retrogressive? With reference to progressive evolution theory, this sterile tissue is required for the fertile spores for the liberation. Okay, for the spreading all over the spores, this tissue is required. This sterile tissue is required. If you see the in the Funaria, you see the sterile tissue in the form of the photosynthetic tissue. In the apophysis, photosynthetic tissue is present, so it provides food also for the growing sporogenous tissue. So as per the this progressive evolution theory, Funaria is the most advanced. It was proposed by Bauer. So next one is, which plants constitute the reed swamp stage of hydrarch or hydrosier? You see, in the hydrosier, you see there are different stages. Okay, free floating stage, phytoplankton stage. Okay, all these are the different stages. Since they are asking about the reed swamp stage, these are also called as amphibian stage, amphibians, because they grow in the marshy areas, swamp areas. Okay, most of they are not immersed in the water, they are not submerged. You see, in the different stages we see there is a submerged stage here, and submerged and free floating stage. Leaves are floating and remaining parts are submerged. So like that, there are different stages here. If you see the reed swamp stage, they are called as amphibians. Examples are Trappa, Sagittaria, Polygonum. So, what they are asked, Typha and Sagittaria. Juncus, Polygonum, not the right options. So, Typha and Sagittaria, this is the right option. So, you have to remember all the different stages of the hydrosphere and xerosphere and their examples. Okay, next question. In which genus glycine is absent in their cell wall peptide bridges? Cell wall peptide bridge. It is a very deep question. The Nocardia, Streptomyces, Frankia, Monospora. So here they come within the bacteria. Okay. In which genus glycine is absent in their cell wall peptide bridges? So Nocardia is the right answer. In the Nocardia cell wall, we don't find the glycine, but in Streptomyces, in Frankia, and in the Monospora, we find the glycine. That is the right answer. And the wall